we have with us coordinator of this workshop, Dr. Subrata Hathi, assistant professor at Dairy Microbiology Department, SMC College of Dairy Science, Kamden University. Distinguished members from BIREC team, Dr. Shilpi Gupta, Dr. Sujit Das, invited eminent speakers, and all the participants who joined us virtually. I, Mittal Kathiria, co-coordinator of this two-day webinar, feel privileged and honored to welcome you all again on behalf of organizing team to the second day of two-day webinar on biomanufacturing of probiotics and fermented foods, a step towards health status and social well-being. Today, we have with us another 3 experienced speakers from academia as well as industry. They are going to speak on different topics related to the theme of the webinar, like regulation for fermented milk products, purification of bioactive peptides from fermented milks, scope and opportunities of fermented milk in Indian market. And then we have open discussion session where all the questions asked by the participants will be answered by the speakers. Dr. J.B. Prajapati sir will act as a coordinator of this session. So I request all the participants to write your questions in the chat box. We'll answer them during open discussion session. Let us start with the first speaker, Shri Aditya Jain, Deputy General Manager in PPT Division at National Dairy Development Board, Anand, Gujarat. He is going to talk on regulations for fermented milk products.
सुजीत तुमको शेयर करो
unmute.
Yes, Mr. Jain is the presenter now. You can share your slide.
जैन साहब आ गया, you can start. आपका अनम्यूट कीजिए जैन साहब, आपका वॉइस नहीं आ रहा है.
हाँ, am I audible now?
Yes, you're audible and visible.
Absolutely.
Screen is also visible, isn't it?
Yes.
Yeah.
Yes.
So a very good morning to all of you. Today I'm going to present on regulations for fermented milk products. First of all, I would like to thank the organizers for the very timely topic and very relevant topic, the SMC College and the BIREC team. Congratulations for this webinar. So let me begin. In my session, what I will be presenting is status of harmonization with Codex Alimentarius, commodity-specific requirements like product description, raw material, permitted ingredients, composition and labeling for name of the food, because these are very specific to particular commodity. And in case of fermented milk, there are certain specificities that need to be seen. And then there are general horizontal requirements. We'll not go into details of those, but we will just see a list of them. So when we talk about status of harmonization with Codex Alimentarius, in 2017, the Food Safety and Standards, Food Product Standards and Food Additives 13th Amendment regulations were published, and they are implemented now. According to them, if we see that all the fermented milk standards have been harmonized fully in principle with the Codex Alimentarius. When we say in principle, means some of these provisions which are not relevant to our country may not have been harmonized and may not have been adopted. And some of the provisions which are very particular to our country have been included. For example, we have added dahi, chakka and srikhand into the fermented milk standards, and we have deleted kefir and kumis because they are rarely manufactured in our country. So as long as the harmonization goes, this standard is totally harmonized with Codex Alimentarius standard for fermented milks. Now, if you see the product description, fermented milk is a milk product which is obtained by fermentation. Of course, that is the crux of the product and crux of the process when it comes to fermented milk, which may have been manufactured using other permitted raw material by action of suitable microorganisms and resulting in lowering of pH with or without coagulation. So it may or may not coagulate because of fermentation, while acidity will go up and the pH will go down. Fermented milk may be heat treated after fermentation, and so there will be some difference in the regulations when it comes to those fermented milk which are heat treated after fermentation, and we will see it later. The raw material used shall be subjected to a heat treatment. So we cannot make fermented milks with raw milk. as per legislation. It has to be subjected to one of the heat treatments which are recognized in the regulations, for example, pasteurization, sterilization, and so on and so forth. Now, when it comes to product description, these fermented milks can be made using some specific starter cultures. For example, dahi or curd. Any lactic acid bacteria can be used for making dahi or curd as a starter culture. When it comes to yogurt, symbiotic cultures of Streptococcus thermophilus and Lactobacillus delbrueckii subspecies bulgaricus have to be used. In case of alternate culture yogurt, Streptococcus thermophilus and other Lactobacillus species can also be used. In case of acidophilus milk, Lactobacillus acidophilus has to be used. And in addition to these, any harmless microorganisms which may not function as starter culture in these products can also be added. For example, we may like to add probiotics or some other functional bacteria that can be added as long as they are harmless. Now, going further in the product description, this standard covers flavored fermented milks, drinks based on fermented milks, and concentrated fermented milks. Flavored fermented milks means a maximum of 50% non-dairy ingredients can be added, including flavorings. So, for example, mango dahi or mango yogurt or similar kinds of products where we are using non-dairy ingredients, maybe we may use dry fruits. So these can be added maximum to the tune of 50%, and the product can be called flavored fermented milk. When it comes to drinks based on fermented milk, these are the products where the fermented milk portion has been diluted with potable water, with or without the addition of whey or other milk and milk products or other permitted non-dairy ingredients and flavors. Here, the requirement is that minimum 40% of the fermented milk portion should be there in the drinks based on fermented milk. And examples are lassi, chach, buttermilk, etc. In case of concentrated fermented milks, the protein content has to be increased prior to or after fermentation, which can be done after drainage of whey or concentrating it beforehand, before the fermentation takes place. It can be done before or after. And product description, it comes to, we can come to the indigenous products like Chakka and Srikhand. They are also included in the in these regulations. Just a minute. So Chakka means, as we all know, partial removal of whey from plain dahi or plain yogurt, and Srikhand means Chakka plus sugar and condiments. So these are all covered under the description of fermented milk products. Yes, some issue. So when it comes to standards, another important component is the raw material that is allowed by the legislation. And in case of fermented milk products, milk, concentrated milk and dried milks, cream, butter oil and anhydrous milk fat as a source of fat, and potable water can be used in case of reconstitution or recombination of, or in case of drinks based on fermented milks. When it comes to permitted ingredients for fermented milks, all the fermented milks can contain starter cultures of harmless microorganisms and other suitable and harmless microorganisms. They can contain salt. They can also be added with prebiotics and probiotics. Sugar is permitted only in case of flavored fermented milks, drinks based on fermented milks, yogurts, dahi, and Srikhand. Nutritive sweetener other than sugar are also permitted, but only in case of flavored fermented milks, drinks based on fermented milks, yogurt, and pre-packaged dahi. So depending upon the requirement of the industry, these decisions were taken to allow or not to allow ingredients. Other non-dairy ingredients such as fruits, vegetables, their products such as juices, purees, pulps, preparations, and preserves derived from such products, cereals and cereal products, coconut and coconut products, honey, chocolate, nuts, coffee, spices, condiments, culinary herbs, and other harmless natural flavoring foods can be used, but only in case of flavored fermented milks or drinks based on fermented milks and Srikhand. Milk and milk products can also be added. That means apart from milk, other milk products, apart from what is given in the raw material, other milk products can also be used in case of drinks based on fermented milks. So for example, if we want to use something to enhance its texture, that can be used. Whey protein concentrates can be used. And starch, it can be also used as per good manufacturing practices and taking into account the use of stabilizers and thickeners, but only in case of fermented milks that are heat treated after fermentation, because they will be required there only. And in case of flavored fermented milks and drinks based on fermented milks, because there you need to add some thickeners. Now, when it comes to composition, the starter microorganisms that are added, the requirement is that they should be viable, active, and abundant. Sum of microorganisms that constitute the starter culture shall not be less than 10⁷ CFU per gram. So 10⁷ CFU per gram is the minimum requirement of starter culture bacteria. Other than starter culture bacteria, if any other bacteria are added, for example, probiotics, they shall not be less than 10⁶ CFU per gram. So in a serving, they will be like, if 100-gram serving is there, they will be like 10⁸ in the serving. And that is what is the requirement for achieving the benefits. If the product is heat treated after fermentation, these requirements for viable microorganisms do not apply. So industry often goes for shelf life extension through heat treatment. In such cases, we cannot claim any benefit of microorganisms or probiotics. So unless otherwise specified, the minimum milk protein content should be 2.9% mass by mass, and minimum titratable acidity should be 0.45% as lactic acid. Now, this is general, but we'll also see that there are certain other requirements for Srikhand, Chakka, and one of the essential manufacturing characteristic in case of fermented milks is that whey removal after fermentation is not permitted in general, except in case of concentrated fermented milks. So only in case you are saying that your product is a concentrated fermented milk, you are allowed to remove some whey after fermentation. Now, in case of plain dahi, the requirement is same minimum percentage of milk fat and milk solids not fat as the case of milk from which it was made. So if you are making toned milk, the plain dahi made from toned milk should have the same fat and SNF percentages as that of toned milk. Standards of dahi prepared from mixed milk will apply if there is no indication. So we'll see when it comes to labeling that in case, just as in case of milk, in case of dahi also, we need to indicate the class of dahi, that whether it is from toned milk or pasteurized milk and so on and so forth. And if we don't indicate any class of dahi, then the standards of dahi made from mixed milk shall apply. Specific compositional requirements have been specified for yogurt, including flavored yogurt and flavored dahi, and partly skimmed yogurt and partly skimmed flavored dahi, and skimmed yogurt and skimmed flavored dahi. Similarly, Chakka, there are specific compositional requirements other than 2.9% fat that we have seen earlier. So for Chakka also there are specific requirements for Chakka, skimmed milk Chakka, and full cream Chakka. Similarly, for Srikhand, there are requirements for Srikhand, full cream Srikhand, and fruit Srikhand. We are not going into the details of those compositional requirements because they are just some numbers and they need to be complied with. When it comes to labeling, the name of the food, because there are certain generic requirements of labeling and then there are specific requirements. So we'll talk about the specific requirements of labeling when it comes to fermented milk products. Every package of food should carry a name which should indicate true nature of the food. So this is general principle of labeling which applies to all the foods, that it should indicate true nature of the food and it should not be misleading to the consumer. In case of fermented milk, the fermented milk may be called in general as fermented milk, or it can be designated with specific names such as dahi, curd, yogurt, or Chakka, Srikhand, or drinks based on fermented milks, depending upon the compliance to relevant provisions. So if you have specified, for example, for dahi, that it should meet the requirement of compositional requirement of milk from which it was made, then we can call it dahi. For yogurt, for example, they have specified some minimum fat and SNF percentages. If they are met, then it can be called yogurt apart from the specific requirements of starter cultures. Now, yogurt or dahi containing non-dairy ingredients, they can be designated as sweetened or flavored yogurt or dahi, because either we will be adding sugar alone or sugar along with other flavoring agents. So as appropriate, depending upon what intervention we have made, we can call it sweetened or flavored yogurt or dahi. The designation of flavored fermented milk shall include the name of the principal flavoring substance or flavored substances used. So if it is mango, you have to indicate it is mango. If it is any other flavor, it has to be very clearly mentioned. Fermented milks to which only nutritive carbohydrate sweeteners have been added may be later labeled as sweetened yogurt or sweetened dahi, or and so on and so forth. Yogurt or dahi containing fruits can be designated as fruit yogurt or fruit dahi as appropriate. Now here I would like to draw your attention to the fact that if you have seen the older regulations, it was not allowed to make flavored dahi. And it was only when we harmonized the standards with Codex Alimentarius and we realized that in case of yogurt, our regulations did permit addition of flavoring ingredients and fruits and all other non-dairy ingredients to make a flavored yogurt or sweetened yogurt. So we thought, why can't industry make, and why are we restricting our industry through legislation? And hence, this was agreed that flavored dahi or sweetened dahi can also be allowed legally. Yogurt or dahi containing fruits may be designated as fruit yogurt or dahi. Products obtained from fermented milk which are heat treated after fermentation can be named as heat treated, and then name of the product, for example, heat treated yogurt or heat treated dahi. And of course, the type of dahi, yogurt, chakka or shrikhand shall always be declared on the label. If it is not declared, then in every case, the regulation specifies a particular category, standards of which will apply. And as we have seen for dahi, it is mixed milk. Similarly, for yogurt, chakka and shrikhand, specific category has been, one of the three categories have been identified, whose standard will apply as a default standard in case nothing is indicated on the label. Additionally, it can have a coined fanciful brand or trade name. So the important thing is you cannot just have a coined fanciful or brand or trade name. It can be additional to the indication of true nature of the food, the name that indicates true nature of the food on the label. In addition to that, it can be there. Apart from this, the label, any label of food has to carry these things: expiry date, date of manufacture, date of packaging. They have to be clubbed together at one place. Then veg, non-veg logo, recommended dietary allowances. So if you are declaring nutritional table, the RDA, percentage RDA met by the product has to be indicated in terms of different nutrients. FSSAI logo and license number, lot code and batch number. I mean, these are all generic requirements that apply to every product: net quantity, retail sale, unit sale price, and consumer care details. Other horizontal requirements that apply to also to fermented milk products are general standard for milk and milk products, including the use of dairy terms. So we have to be very mindful that when we are calling a product by a name that is understood or that is recognized as a dairy term, for example, dahi or yogurt or chakka or shrikhand, then there's a standard called use of dairy terms or application of dairy terms, and it is under the general standard for the milk and milk products, and those provisions apply. Then there are provisions for food additives, provisions for contaminants, toxins, and residues, hygiene requirements, and methods of sampling and analysis. So when it comes to the use of dairy terms, because this is becoming important nowadays, I would like to share that any word, pictorial representation, graphic representation, or any impression on the label or labeling information that is suggestive of or that gives an impression that it is a dairy product to the consumer, they are called dairy terms. So if you, for example, if we call some product milk, so what comes to our mind is that milk is something which is drawn from animal and it should be pure and it should be, nothing should be added, nothing should be withdrawn from it. So such kind of terminologies cannot be used for any products which are not milk products or composite milk products. Composite milk products means products such as flavored yogurt and flavored dahi, etc., which have non-dairy ingredients, but they are not intended to replace the dairy portion of the product. Apart from products such as analogs, in which the non-dairy ingredient is intended to replace the dairy ingredient, dairy component. So, for example, if fat is replaced with a non-dairy fat in case of yogurt, then you cannot call it yogurt. You have to use some other terminology. So, just to see that in a little briefly, to see how the use of dairy terms is allowed in India. So, in case of milk, the term milk can be used for milk, as defined, or milk which is adjusted for milk fat or milk solids not fat content. So we can use the term milk for milk as it is drawn from the udder, or also for the milks that are allowed in the legislation, such as toned milk, standardized milk. So they are adjusted for milk fat or SNF content, but they can still be called milk. Milk products, dairy terms can be used for a product which is qualifying as a milk product or a milk product with fat, protein, or both adjusted while meeting the compositional criteria. So there is a compositional criteria, for example, given for yogurt as 2.9% fat. So as long as we are adjusting the milk fat within that range, it can be called yogurt. But if we go beyond that, or we reduce the milk fat content to less than that and add some other fat, then it cannot be called yogurt. And a milk product which is modified while retaining its essential characteristics, we can still use dairy terms. For example, lactose-reduced milk and milk products. They are still milk products. In case of composite milk products, dairy terms can be used with a clear description of other characterizing agents, such as flavoring foods, spices, herbs, flavors, etc. So, for example, if you have yogurt added with mango, it becomes a composite milk product, where mango is actually being used as an ingredient for imparting flavor. It's not to replace milk and milk components. So there you can use the term yogurt, but along with the characterizing description of the characterizing ingredients, such as mango. Then in case of imitation or analogs, where any of the components, whether it's milk fat or milk protein or any other ingredients or any other constituent of milk, that is replaced with a non-milk constituent. So if it is non-milk fat being added after removal of milk fat, or non-milk protein being added after removal of milk protein, so sort of replacement, if it happens, then dairy terms cannot be used. So for example, I am showing a picture here, which shows almond yogurt, and it contains 23 gram protein from almond. So this cannot be called actually yogurt. So it's very confusing to the consumer, and it's not legally acceptable. So such products cannot be called by using any dairy terms. So with this, I think as long as the regulations on fermented milk goes, I would like to finish my presentation. If you have any questions, I can take it, or maybe if the organizers have decided to take the questions at the end, we can take them at the end. Thank you.
Thank you so much, Aditya sir. It was really a very informative presentation. Our next speaker is Dr. Subrata Hati. Sir is Assistant Professor at Dairy Microbiology Department, SMC College of Dairy Science, Kamden University, and he is also the coordinator of this workshop, this webinar. Sir is going to talk on the topic production and purification of bioactive peptides from fermented milks.
One second, sir.
Participants, kindly write your questions in the chat box. We'll answer them during open discussion session.
And a few of them I've replied in the question box also. That will take up your questions at the end, like Dr. Mittal has said. There's a couple of questions which are coming, but we'll take them up one by one at 12:30.
Sujit, my screen is visible?
Absolutely. It's clear, sir.
Okay.
Fine.
Voice is also clear, right?
Yes, absolutely.
Okay. So good morning, all the participants. And also I'm very much thankful to the BIRAC team for selecting our college, our department, for organizing this 2 days webinar on a very important topic on fermented milks and probiotics. And I also welcome all these learned speakers, previous days speakers, as well as present day speakers. Now I will present my topic on production and purification of bioactive peptides from fermented milks. So I will talk about the milk protein, because we are working here in our dairy microbiology department, mostly the milk and milk products. So milk, how we'll extract the bioactive peptides or the peptides from the milk protein, that is very important for us. So milk proteins exert a wide range of nutritional, functional, and biological activities. Physiologically active peptides are mostly derived from the milk proteins. These peptides are inactive within the sequence of the parent protein molecule. It is released during the gastric digestions or during the food processings. If we take the example of cheese, then when we are doing the renneting, rennet is an enzyme, then there is a chances of producing the milk peptides from the caseins and the whey proteins. Basically, the bioactive peptides generally contain 3 to 20 amino acid residues per molecule. So basically, the bioactive peptides, it's a small amino acid residues compared to the long polymer chain of the proteins. Many bioactive peptides possesses multifunctional properties. I will discuss in the later slides. At present, milk proteins are the most important source of the bioactive peptides, and most of the studies are being done extensively on the milk proteins of different species also. A variety of naturally formed bioactive peptides have been found in fermented dairy products like yogurt, dahi, lassi, even probiotic dairy foods or probiotic fermented milks, cheese. Bioactive peptides are released upon the fermentation of milk using different live proteolytic microorganisms or proteolytic enzymes derived from such microorganisms. So basically here, this term proteolytic is very important when you are talking about the peptides, because these organisms produce a lot of proteolytic enzymes like peptidase, aminopeptidase, proteases, proteinases. So these are the different enzymes basically produced by these proteolytic microorganisms during the fermentation or during their growth in the product. And these enzymes basically act on the milk proteins, and then the milk proteins are being fragmented into smaller chain of the amino acid residues, and then it is converted to the small fragments of the peptides, and their length is in between 3 to 20 amino acids. Fermentation of milk with a certain dairy starters, peptides with various bioactivities can be formed and detected in an active form even in the final product, and they can be absorbed and reach the peripheral organs by using different transporters. So milk fermented with Lactobacillus helveticus, MTCC 5463, that is our own indigenous cultures. We have worked on a lot on these cultures. It is a clinically proven probiotic cultures, and it has a good proteolytic activity also. So we have prepared a fermented milk with this Lactobacillus helveticus, which have given good amount of ACE inhibitory activity and ACE inhibitory peptides during the fermentation process. In the gastrointestinal tract, digestive enzymes may further degrade long oligopeptides, leading to a possible release of the bioactive peptides. Once liberated in the intestine, bioactive peptides may act locally or pass through the intestinal wall into the blood circulation, ending up in a target organ with a subsequent regulation of physiological conditions. So here, basically, any bioactive compounds produced by the food materials, they are being transported to the blood circulation systems using a different transporters. Similar for the peptides also, when it is released into the intestine or it is carrying by the food matrix, so they are being transported into the blood circulation systems, and they goes to the target site, and then we get some benefits from these peptides. So the bioavailability and biostability is a very important part of the bioactive peptides, depending on their amino acids, peptides, or the characteristics of the amino acids. Maybe it's a hydrophobic, hydrophilic, aromatic. So what kind of amino acids are present in the peptides, and their concentrations, their order of the sequence is very much important for the peptides when they are getting absorbed in our intestine or they are being transferred into the peripheral organs. So bioactive peptides derived from the milk proteins, I already discussed that. Basically, we are in dairy microbiology field, so we work in extensively on the milk and milk products. So you know the milk has two different proteins, caseins and whey protein. Casein is the major protein of the milk, and whey protein is the smaller one. 80% is the casein and 20% the whey protein. Under these caseins, there are different fragments of the proteins like alpha is 1 casein, alpha is 2 casein, beta casein, kappa caseins. And you can see here that alpha is 1 and beta casein are the major protein portion of the caseins, and protein beta lactoglobulin is the major contributor of the whey protein out of alpha lactoglobulin, beta lactoglobulin, bovine serum albumin, and immunoglobulin. So this is the distribution of the milk proteins in a fragmented way, that what kind of proteins are present in the milk. Now comes to the bioactive peptides. Proteins in foods do not only serve as nutrients, but also perform the physiochemical roles that promote various health benefits. And bioactive peptides are organic substances formed by the amino acids joined by the covalent bonds, and also known as amide or peptide bonds. Bioactive peptides are structurally short-chain form of amino acid residues around 2 to 20, and their molecular weight is less than 6,000 kilodalton. So you can understand that the peptides are basically a protein, but it's a smaller fragment of the protein, because protein is a long polymer chain of the amino acids. So basically, the enzymes are breaking the proteins at different amino acids site, and they create these peptides. So bioactive peptides are specific protein fragments that have a positive impact on functioning or conditions of the living beings, thereby improving the health. Bioactive peptides are inactive within the sequence of the parent protein, but after the release by enzymatic hydrolysis, exact various physiological functions. The bioactivity and functionality of peptides depend on the amino acid composition, sequence, and molecular masses. And bioactive peptides are known for their ability to inhibit the protein-protein interactions. More than 1,500 different bioactive peptides have been reported in the database named the BioPep. So basically, I already discussed that the characteristics and the structure and bioavailability, also the biostability, depend on the composition of the amino acids, sequence of the amino acids, and the molecular masses of that particular protein or the amino acids. So bioactive peptides are known their ability to inhibit the protein-protein interactions. We have worked on it. Some molecular docking we have started, where these different enzymes cause some oxidations or other cellular damage, but the peptides are able to bind them by using different ligands. Until date, there are 1,500 bioactive peptides are started and reported in the BioPep database, and they are being extracted and studied from the different food proteins. So if we see the sources of the bioactive peptides, from plant sources, as we eat rice, wheat, corn, maize, and barley. Then for animal sources, fish, chicken, beef, and eggs, and milk, which I will talk about. The milk, basically, we use the fermentation of the milk, like yogurt, cheese, and we can also go for some hydrolyzers using some proteolytic enzymes like alkalase, papain. So these kind of enzymes are also being used to produce the milk hydrolyzers from casein or from whey proteins. Vegetables also are a rich source of the protein, and they can produce the peptides like spinach, pumpkins, mushroom, green peas. If we see the market status of the bioactive peptides, according to the Cohort Market Insight 2019, the global bioactive peptide market size was valued at $3,265.2 million in 2017, and they're expected to exhibit a compound annual growth rate of 9.4% over the focus period between 2018 to 2006. And North America is dominating the global bioactive peptides market in 2017 with 35.9% market share in terms of the revenue. And these are the players who are selling the bioactive peptide-rich products, like Arca Daniels from US, Seagarden from Norway, Pumpep from China, NatureDate from US, Royal DSM Netherlands. So most of the Western countries, they have, even China, even Japan also, they are selling the bioactive peptide-rich product as a functional food or as a health foods in different metrics, by using different food metrics like milk or other food metrics. How the bioactive peptides are synthesized? It can be synthesized chemically. it can be synthesized through enzymatic hydrolysis or through the microbial fermentations. And when you eat any foods, when it reaches to the intestines or the stomach, there are a lot of proteolytic enzymes or enzymes are there, and they can degrade these proteins and they can produce peptides in through gastrointestinal digestions. So how these peptides are being produced and what are the factors we should consider when we are producing the bioactive peptides? First is the amino acid sequence of the protein substrates. As I already told, the length of the peptides, the molecular weight of the peptides, the charge of the peptides, or the amino acid residues of the peptides are very important. In pre-treatment, we give some pre-treatment to to enhance the work of the enzyme. So pre-treatment is required. Enzyme specificity, what kind of enzyme, you have to screen out the enzymes, which enzymes will be better for that kind of protein, because there are a lot of proteolytic enzymes are there, but you have to screen it out on the basis of your requirement, that which enzyme will be the suitable for production of this peptide at optimum level. And then the enzyme act at a particular conditions, hydrolysis conditions like pH, temperature. So you have to also standardize or optimize that conditions. Then after production of the peptides, you have to inactivate that enzyme by giving heat treatment or other ways. Then we have to go for downstream processing like separation and purification. This is the basic flow diagram, or this is the basic protocol you have to follow for the production of peptides from any food proteins. More than 60 peptides already being approved by the US FDA as a medicines on the market, and this is expected to grow significantly with approximately 140 peptide drugs currently in the clinical trials and more than 500 therapeutic peptides in the preclinical development. As I already told that how the peptides are being produced. Here you can see that the peptides are produced with the help of the digestive enzymes like proteolytic enzymes, or through the hydrolysis by the proteolytic enzymes like pepsin and trypsin or alkalase, and through fermentation by using starter cultures, probiotics. Okay, so we can use the organisms during the fermentation process, or we can digest these food proteins through in vitro digestions. We can create a conditions where we can digest the proteins, and then these proteins will be converted into the peptides, and that peptides can be added into the food products for different formulations, and also through the enzymatic hydrolysis. And these peptides will give you different health benefits like vasoactive, cytomodulatory, mineral binding, opioid activities, antioxidative, immunomodulatory, antimicrobial, and antihypertensive activities. So these peptides will give you different health beneficial properties. Then production of bioactive peptides from whey protein. So here I have shown you one basic process or the method through which you can produce the peptide by using the enzymes. So I have taken an example of whey proteins. That work is already being done in our lab also. I have taken the example from the NDRI. 5% whey protein concentrate solutions they have prepared. They have used 80% WPC. From that 80% WPC, they prepared 5% WPC solutions, and they heated at 68 degrees Celsius for 10 minutes. After heating, then they hydrolyze by using these three different enzymes: alkalase, chorolase, and flavorzyme. And they have optimized these different conditions like enzyme substrate ratio, pH, time for the hydrolysis, and temperature. So these are the parameters they have optimized for the maximum hydrolysis and maximum production of the peptides. After that, they have inactivated the activity of the enzyme by heating at 90 degrees Celsius for 10 minutes. Then they have started the downstream processing for purifications of the peptides by centrifugations. And after centrifugation, they pass through different cutoff membranes like 3, 5, 10. Also, you can choose another ultrafiltration systems for less than 1 kDa even. Then after passing these hydrolysates, they pass through different cutoff membranes, and then they go for the different techniques to identify or characterize the sequence of the peptides through LCMS or MALDI-TOF. There are other advanced techniques are there where you can identify the sequence of the peptides. People have already worked on different fermented milks, and they have produced the bioactive peptides. they have used single organisms or in a combinations of the organisms, or they have used both organism plus enzymes, means two-stage fermentations. So here you can see the Lactobacillus helveticus along with the yeast Saccharomyces cerevisiae. They have used and they have fermented the milk. Basically, the source of the peptides are from beta casein and alpha caseins. The peptide sequence are valine, proline, proline, and isolation proline, proline. And these tripeptides have a ACE inhibitory activity or anti-hypertensive activity. Another example they have given from the Lactobacillus GG. Basically, it is a Lactobacillus rhamnosus GG, and they have used two different enzymes, pepsin and trypsins, and they have produced different fragments of the peptides from these two major milk proteins, beta casein and alpha S1 caseins. And these peptides have opioid activity, ACE inhibitory activity, immunostimulating activity. Similarly, they have also used Lactobacillus rhamnosus plus pepsin and corolase from beta caseins, and they produce 3-4 different peptide fragments, and that fragments have a ACE inhibitory activity as well as the antioxidative activity. They have also used the single organisms like Lactobacillus helveticus, CP90 proteinase, Lactobacillus helveticus CP94, Lactobacillus delbrueckii subspecies bulgaricus. So these, you can see that different fragments of the peptides are being generated from the beta casein and alpha caseins, basically from the casein part of the milk protein. And here you can see that whey protein also they have generated the peptides. This is dipeptides, tyrosine and proline, and they are having good ACE inhibitory or antihypertensive activity. What we are working at Anand on bioactive peptides, we have started in 2013-14. So we have started the bioactive peptide production from the camel milk, and we want to see the antihypertensive effect of the camel milk by fermenting with our own lactobacillus cultures. So that work we have started more than around 10 years back. These are the cultures we have considered at the very beginning for this study, mostly lactobacillus and 1 or 2 other organisms, Streptococcus thermophilus and Lactococcus lactis. These organisms were considered for the production of the peptides and fermentations. So we have also estimated the ACE inhibitory activity of these organisms during their fermentations, and we have found out that NS4 and NK6 are giving the best results. Even 09 also, they're giving maximum ACE inhibitory or anti-hypertensive activity during the fermentation period. We have also checked or evaluated the peptide production by passing through different cut-off membranes, and LVF is giving the maximum peptide production around 65% at 10 kDa per minute. For NS4, it is around 56%. Then we have checked the protein profile of the fermented camel milk, which is being revealed here by SDS page analysis. So you can see over there that peptides are being fermented with these cultures, and they are showing a lot of bands. But after fermentation, the bands are getting disappeared because of the hydrolysis of the proteins with the strong proteolytic enzymes produced by the bacteria during fermentations. Then we have purified those peptides through the 2D page, and these pods were trypsin digested, and we have done sequencing through LC-MS. And these are the 3-4 sequences we have found out from the camel milk, GPPY, QP, LBPR, and other 3. They are basically sourced from the beta casein and alpha S1 casein and alpha lactoalbumin. And these sequences we have submitted to the NCBI and also in the BioPep database, because this is our own indigenous peptides from the camel milk. This is the Kutchi breed of the camel from Gujarat. So when we started the work, we were a bit because we did not have this facility to use the peptide sequences already being done in our country. So we have started work in 2014, and on that time we did not find any kind of sequences already being submitted in the databases. So we have done this job. We have submitted it into the BioPep databases. Now people can use our sequences for seeing or for matching with their own sequences that this is from the camel milk. We have also done the anti-inflammatory activity of the fermented camel milk in the raw macrophages cell line. And you can see over here that the cell viability is increasing at 0.25 milligram per ml fermented camel milk concentrations. And we have checked the nitric oxide productions and other anti-inflammatory markers like TNF alpha, IL-6, and IL-1 beta. And you can see that the culture KGL4 with the fermented with the camel milk can reduce the TNF alpha production, IL-6 productions, and IL-1 beta production compared to the LPS induced raw macrophage cell lines. And when we are giving with in combination KGL4 plus LPS, again compared with the LPS lipopolysaccharides, the reduction was quite significant. So we can see that the fermented camel milk with KGL4, that is lactobacillus fermentum, is giving the best results in controlling the anti-inflammations. We have also produced fermented camel milk peptides by using both lactobacillus as well as the yeast Saccharomyces cerevisiae. These are the 6 different camel milk peptides we have isolated, and we have characterized, and then we have deposited in the BioPep. We have also worked on ACE inhibitory peptides from these camel milk, as well as we have also worked in the antioxidative activity of the camel milk as well as the goat milk. Here we have worked on ACE inhibitory activity of fermented goat milk. So we have used 5 different lactobacillus cultures for the fermentation of the goat milk. It is a Shruti breed from Gujarat. So we heated the milk at 90 degrees Celsius for 10 to 15 minutes. Then we have cooled at 37 degrees Celsius. Then we have added the cultures at the rate of 2%. And then we have collected the sample at different intervals, 0, 6, 12, 24, and 48 hours. And we have checked the different antioxidative activities also. Then we have also seen the ACE inhibitory activity of the lactobacillus culture during fermentation of the goat milk. So these are the peptides we have found out from the goat milk. And this is the SDS page protein profile of that goat milk fermented with these lactobacillus cultures. We have also already I have shown that we are working in antioxidative activity of the fermented goat milk. So these are the 3 different assays: ABTS assay, hydroxyl free radical scavenging assay, superoxide free radical scavenging assay. These assays we have used for the checking of antioxidative activity of the fermented goat milk. So you can see over here the results that as we are increasing the fermentations, the activity of antioxidant or hydroxyl free radical scavenging activity, superoxide free radicals, these are increasing. And if we see the peptide production also, as we are increasing the fermentations or rate of inoculation, the peptide production are also increasing. And we have also checked their protein profile on the SDS page. And after doing 2D, we got these sequences from the fermented goat milk by our M4 cultures. So these are the sequences which are matched with the PIR and BioPep databases. And these peptide sequences have a antioxidative activity by comparing with the BioPep databases. We have also started to work in the sheep milk. We have extracted the peptides which are having antioxidative and antimicrobial activity. So these are the peptides are being extracted from the sheep milk. This is called a Panchali breed. It is very popular in Gujarat. So we have taken the milk from this breed and then we fermented with our own cultures. And after fermentation, we have extracted or purified the peptides from this fermented sheep milk. Basically, here we have done with KGL4 cultures, lactobacillus fermentum. And then we have done the SDS2D. And also we have checked the molecular interaction study of the peptides, which have a peptide ranking score is more than 9. And that peptides have the capacity to bind these enzymes which cause the oxidative damage at a cellular level. So it's a in silico analysis of that peptides, that how they can bind those enzymes which cause the damage to the cellular level, and they can stop the reaction of the enzyme so that we can manage the oxidative damage to the cells. We have also checked the anti-inflammations by using different anti-inflammatory different markers like TNF-alpha, IL-6, and IL-1 beta. Also, we have checked the reduction of the nitric oxide production of fermented sheep milk with KGL4. We have developed a product which is rich with peptides. We have added WPC, whey protein concentrate 80, and calcium caseinate. And then we have fermented with lactobacillus cultures, Lactobacillus helveticus. And these we have added a different concentration of the proteins, 1%, 1.5% of WPC, and 2% calcium caseinate. And then we go for the sensory and other attributes, microbial attributes, as well as the ACE inhibitory activity of this product during the storage study. And we have found good amount of protein is there, and peptides are being released from the proteins which are having a ACE inhibitory activity. So people have already developed some products which are rich with different peptides. The sheep milk feta cheese, sheep pecorino cheese, sheep Roquefort cheese, sheep milk yogurt, goat milk kefir, goat milk kefir. So these are the different fermented products with different milk like sheep or goat. And these are the different peptides they have identified from the beta caseins, beta lactoglobulin, and kappa caseins. And these are the activities they have found from these peptides which are rich with these different fermented milk products. Some of the studies I have collected from different literatures that where they have developed a product rich with the particular peptides, milk fermented with Lactobacillus helveticus and Saccharomyces cerevisiae. So here you can see two organisms, Lactobacillus helveticus and Saccharomyces cerevisiae. So yeast as well as Lactobacillus in combinations, they have fermented the milk, and that milk are basically rich with BPP and IPP, valine, proline, proline, and isolation proline, proline. They are derived from the beta casein and kappa casein respectively. And then, then they have given this fermented milk to the single-blinded placebo control trial to the 30 subjects with mild hypertension. And every day they have given 95 ml of that fermented milk, and that milk is rich with 1.1 milligram IPP and 1.5 milligram BPP. These two peptides are they have quantified in the particular dose, and they've given for 8 weeks. And what they observed after giving them for 8 weeks, that 14.1 millimole Ag reduction in the systolic blood pressures and 8.8 reduction in the diastolic blood pressure. So they have got a significant results when these peptides are present in quite rich amount in the particular dose. Another study, they have prepared yogurt enriched with casein hydrolyzed. A yogurt enriched with the casein hydrolyzed containing alpha-1 casein derived antihypertensive peptides, and these two peptides are there in this yogurt, RYLGY and AYFYPEL. These two peptides are there in the casein hydrolyzates, which are being added into the yogurt. And the study was conducted for normalized placebo control trial in 71 subjects with a mild hypertensive they have, and they have given 20 ml per day, and that dose means these 20 ml contains 5.5 milligram of RYLGY and AYFYPEL. These two peptides, 5.5 milligram amount, they are present into the per dose, and that the study was conducted, or the dose has been given through that subjects, 71 subjects for 6 weeks, and they have found that 12.5 mAg reduction in the systolic blood pressures. Another studies on fermented milk enriched with whey protein concentrate. The composition of pasteurized milk, they have done the formulation: skim milk powder 6.6%, WPC 80%, WPC 2.2%, and for placebo WPC is 0, means they have not added in WPC. Instead of WPC, they have added in soybean peptide 0.26%, sucrose 0.7%, fructose 0.7%, and sweetener 1.02%, flavoring agent 0.09%, and water around 80.43. So around 18 to 20%, 16 to 18% total solids are there. Then they fermented that pasteurized milk with Lactobacillus casei and Streptococcus thermophilus, and they have added these cultures in 6.1 into 10 to the power 8 CFU per ml and 2.6 into 10 to the power 7 CFU per ml. And after fermentation, they made this fermented milk product which are rich with the peptides, because they have added WPC as well as the soybean peptide. And study was conducted for single blind parallel placebo controlled trial in 20 healthy men. And their dose level was 200 ml for 2 times a day for 8 weeks. So it's a huge amount of milk they have consumed during the studies. 200 ml for 2 times means 400 ml a day. The result, what they observed, 7 mmHg reduction in the systolic blood pressures versus the placebo. So there is a reduction. Even in the graph, you can see that as the study is going towards the end, there is a significant reduction in the systolic blood pressures from different clinical subjects. People have already developed different fermented or peptide-rich dairy products like Gouda cheese, Emmental, Camembert, Havarti. So these are different cheeses they have already produced or manufactured with different fragments of these peptides. That most of the peptides are from, you see the caseins, alpha-1 casein, beta caseins, even yogurt, BPP and IPP, dahi, SK, VYP, caprine kefir, then ovine cheese, and also another kefir. So they have produced these different fermented milks with different fragments of the peptides. Now, commercially available products are there or not? Yes, it is there. Japan was the first inventor of this peptide-rich product. It's a very popular product in Japan that is called Calpis. It is rich with the VPP and IPP, and I have already told that these products are being manufactured or prepared with 2 organisms in combinations, Lactobacillus and Lactobacillus helveticus and Saccharomyces cerevisiae. So here they have used 2 organisms in a combinations, and they have produced good amount of antihypertensive. Basically, this Calpis is a antihypertensive peptides. Then another product is peptide C12. It is basically the product produced by the company DMB International Netherlands, and their peptide source is the casein hydrolysates. This is the sequence of the peptides. Then Evolas, it is also a very popular product, quite old product on peptide-rich product. And this is the company who produced this product, Valio Oy, Finland. And this is a calcium in this fermenter, so they have added the calcium also. Then Lopept, it is produced by the Inobs Biotech Spain. Their peptide source is the casein hydrolysates. Then amyl peptide, Calpis company Japan, they have another product, amyl peptides. Basically, it is a beverage mixture of vegetables and fruit juices. So those who don't take milk products, they can take this one. Casein DP Pepto drink, Kanebo, Tokyo, Japan, they have another product on peptide-rich product for different health beneficial properties. So these are the functional foods or healthy foods are already available in the market. Most of the companies, they have already tried it, but one thing is that they have done clinical trials. They have proved that that amount of peptides with a particular dose for a particular days, you get some kind of benefits. Means you can manage your health if you are hypertensive or if you have some other health problems. So these products are clinically proven. Now I am concluding my talk here with a few recommendation like statement. Bioactive peptides offer a new means to promote the health and can provide health benefits beyond their basic nutritional role. There is a tremendous global interest in promoting the use of food proteins, peptides, as a novel alternatives for present pharmaceutical therapeutics in the treatment and prevention of high blood pressure and other lifestyle diseases. And milk peptides are the most studied bioactive peptides, and various health beneficial properties have also been studied. So thank you all for listening me. So I'm working here in our department. We have our colleagues, Dr. Mittal, Dr. Srija Madam. So we're working together here, but we have some collaboration internally. Dr. Amor Sakure from Anand Agriculture University, Dr. Prakash Karinga from our own university, and Dr. Nikas from Poultry Science Department, Karinga from Animal Biotechnology. And our external co, Dr. Kanti, is my good friend. He's helped a lot. Dr. B.K. Misra from Nehu, Silong, and Dr. Pritam from NIT Raurkela. So with this, I am concluding here. Thank you very much.
Thank you very much, Subrata sir. You have started with basic concept of bioactive peptides. You showed us various processes for production of bioactive peptides. You also shared many research studies for the production of bioactive peptides from different fermented milk obtained from the various species. You also shared commercially available bioactive peptides. So it was really very knowledge enriching presentation. Thank you. Thank you for that, sir. Our next eminent speaker is from industry, Mr. Samir Saxena. Sir is head at Quality Assurance and New Product Development Division at Gujarat Cooperative Milk Marketing Federation, Gujarat. And today he is going to speak on the topic, Scope and Opportunities of Fermented Milks in Indian Market.
Hello.
Yes, sir.
Yes.
Am I
Hello,
audible?
sir. Yes, you are audible, sir.
Yes, sir.
Okay. I'm just trying to share my presentation. Just hold on for a minute.
Sujit, make them
Yes, we have-
presenter.
Yes, we have made Saxena sir as the presenter. You can share your slide.
Show my screen.
Yes, it is visible, sir. You can put your presentation in the slideshow mode.
Put your slide, sir. Your screen is visible.
Okay. Let me see. Yeah, is it visible now?
Yeah, absolutely.
Yes, sir.
Yes.
Yes, sir.
Okay. So, good morning, everyone. I'm Samir from Amul Group, and I would like to thank Birak for inviting me as one of the guest speakers. I will showcase you what the products are there in our basket, fermented products and probiotics. So I'm actually pressed for some time because we are all at Amul involved in arrangements of some function with the PM. So I'll try to finish as early as possible, but I'll see that I drive home what I want to. Hello.
Sir, you
We can
are
hear you,
audible,
sir.
sir. You continue,
You're
sir.
Audible. Okay.
You are
Please
perfect.
let me know if there is any distraction. I'm starting my presentation. Okay.
Please.
So
Yeah.
this is something to tell you about Amul. You must be knowing that we are a group of 3.6 million farmers who are our owners. They are basically poor, landless, and marginal farmers associated with us. And I see some disturbances in between. Anyway, so we are collecting almost 2.7 million liters of milk every day to 3.6 million farmers, not only in Gujarat, but also in 18 states outside Gujarat. We are the 8th largest dairy processor in the world and having a total capacity of 41 million liters of milk handled every day. And we are basically now 9 CAGR of 16%. Okay, so this is actually our philosophy: value for money and value for many. So value for many means we the 3.6 million farmers. If the whole dairy business is of 1 rupee, then 85 paisa goes back to our farmers. So 80 to 85% of the money involved in the dairy business goes back to the farmers. We have a very strong supply chain divided into 4 components. One is the frozen, chilled, ambient, and fresh. Yeah. So as per IFSCN, we rank the 8th in the world as far as dairy processor is concerned in top 20 dairy processors. Okay. Now, as per the Kantar report, we stand third largest globally picked up brand from the shelves. That is 6 billion times our packets are picked up, not counting the market milk which we are dispatching on day-to-day basis. Almost 150 lakh liters of milk is sold every day. So if we count that number also, then we are number one in the world as the most picked up brand. Then as for the Rabobank ranking, we stand 13 as far as the turnover. Now coming to the trends which are existing in the world and India, that the global fermented milk products market is growing globally by a rate of 1.5%. Global probiotic fermented market is 3.7%, whereas in India, the growth of fermented foods like yogurt and dahi is growing at a rate of 15.3%. And this is, we stand fourth in the world as far as fermented products are. We are number 4. Now, these are the key drivers of fermented milk products, which drive the sales of the fermented products or probiotic products in India. One is the increasing health awareness, then diverse product offerings. You will see the whole litany list of the products we have in our basket. Then urbanization and changing styles, lifestyles, rising disposable income, rural market potential is there. We have started marketing our products, milk and fermented products, also in the rural markets and 10K towns. Then innovation and product development, retail expansion has taken place a lot in India, and government initiatives. Government is providing PLI schemes for the industries to encourage the industry to go ahead with the production of fermented products. These are the consumer trends which drive the sale of the fermented products like healthy lifestyle, convenience, and functional foods. Okay, then the scope of fermented products and probiotic products, is rising because every day, then we are offering fermented products for the masses at a very affordable prices. Probiotics for the masses, you will see what products are there in our basket. Then the regional fermented products are catching up in India. We are also targeting exports and trying to minimize the imports of fermented products. Then there has been a shift from loose to packaged products during the COVID. We are targeting millennials and Generation Z for fermented products. Now they are getting educated and getting galvanized for fermented and probiotic. Probiotic products are now being used as health supplements. Then there are specific products like high protein yogurts and yogurt breakfasts in India. And we are also now on the journey of start making the cultures also in India. So that we make in India first. These are some of the categories which FSSAI has already laid for fermented products like fermented milk, drink based on fermented milk, concentrated fermented milk, plain dahi, and flavored fermented milks. The categorization of yogurt has also been regularized by FSSAI. Now these are some of the products which we have already launched and they are there in the market. Like this is Masti Dahi in various shapes and sizes and denominations, 85 grams, 200 grams, 400 grams. Then dahi also is in pouches. Low-fat dahi, kadhi dahi. This is very sour dahi intended for making kadhi at home. Then this is a set yogurt in strawberry on the basket. Then we have got stirred fruit yogurts with inclusions, which we have recently launched last year. So they are available in 4 flavors. Then we have got some regional preferences like Meetha Dahi. This goes very well in UP, Mishti Doi in West Bengal, and Sweet Lassi. We are doing it almost pan-India. Then this is Kathiawadi Chhat, we call it. It is very sour buttermilk, which is live very much in Saurashtra region of Gujarat. So that is also there in pouches at a very, very affordable. Other variants like Amul Sattva Chhat is also there. This is having some probiotic bacteria. Sour buttermilk. This is, the high protein fermented buttermilk packed in Tetra Pak with a long shelf life, which was showcased during International Dairy Federation last year, and we received the award for these two products. Then we have got high protein products like blueberry shake of protein per 200 ml. Then we have got high protein lassi in Tetra Pak, giving 15 grams of protein per pack. Then this is high protein dahi, 25 grams per 400 grams of cup. Is already there in the market on Amul shop. You can find it. You can purchase it. Then this is very shortly we'll be launching high protein yogurt, pineapple flavored, 30 grams and 200 gram pack. Then Shrikhand is a very traditionally old product which we are also doing currently. And we have got sour cream and some dahi based savory items are also there on our cart. Panchamrit we had launched some 5 years back. Now is the time when you see that there is a lot of religious places getting momentum like Ayodhya, then Kashi Vishwanath and Mathura. The whole corridor is now developing up. So this product we had launched 4 years back and is now doing well. And it was also distributed recently during Ram Mandir Pran Pratishtha. Along with Big Basket, we sold it at ₹1 for the consumers to get the high awareness of this product. Then if you look to the probiotic products present in India, they are very prohibitive, done by some MNCs because of the price they have, the promotional expenses they do, and a very high sugar content. So against that, we have launched a product which is affordable for the masses. This is probiotic buttermilk in pouches. 30 rupees per liter. And a common man also can have a probiotic goodness of probiotic bacteria through this product. We are selling almost 15 lakh liters of buttermilk every day. So you can imagine we are almost in a family. If we have 4, so we are touching almost 60 lakh people every day through this mass product at a very affordable prices. We had advertised this in the newspapers, local newspapers. Then we have got a probiotic range in frozen products like Amul Chocobar is probiotic. If you pick up, find it is probiotic and it is being sold pan-India. Then some frozen yogurts are there on the card. Then probiotic range in fresh products like we have got probiotic dahi, probiotic buttermilk, probiotic lassi, yogurts, and as I said, probiotic buttermilk is also there for the masses. Then these are some new urban market packs available in Mumbai and South. Then we also launched probiotic chocolate. This chocolate has probiotic bacteria goodness. Then way back in 2007, actually our journey with probiotics started way back in 2002, and we launched this Amul Pro Life probiotic ice cream in 2007. Thinking that the cultures you have to store at -18, and this ice cream is also to be distributed at -18. So the probiotic ice cream would be the best vehicle for transportation to long places. That's ice creams. We were the first ones to do it in the country. These were the health campaigns which were there on the media, newspapers. Then we did some promotional activities on radios and banners. Visibility at point of sales were also done for this product, and we received the first prize for Amul sugar-free probiotic ice creams in 2007, way back in 2000. If you Google for Chocobar, you'll find Amul stands first in the Google search, which is probiotic. Then we have done a lot of promotional activities to promote our probiotic products for sampling. Then TV campaigns. I'll see if one of the campaign runs. Yeah, something like social media, TV, then a few ads like this. Social media campaigns. Then on the social media, we were also, showcasing our different products, fermented products. Then targeted advertising like Durga Puja and some events like the football World Cup. Then some regional language promotion was also done in Marathi, Punjabi, Gujarati, Bengali. And this is the latest one in Telugu. I'll just run it for you. So such type of ads will come in other languages also. So this was the first attempt in regional languages. Then we have through our social media of Facebook, YouTube, Twitter, and likewise. Then very recently, just a month back or so, we have started exporting our Masti Dahi to Singapore in various denominations like 200 ml, 400 ml cup is there, 1 kg, and 2 kg buckets are there. So 3 consignments have already gone, and we are catering to Singapore market since last a month or so. Then we are also on the journey. We are trying to do it. Hopefully, we'll be successful in this endeavor. And we are also now thinking for probiotics for animals. We have looking for some probiotic bacteria for animals made in India. But definitely, we have conducted some trials based on the imported cultures, and we see that there is a good improvement in the animal health if we give probiotics to the animals. Okay, so this is all I have. Thank you.
Thank you very much, Samir sir.
Yeah.
We know you are very busy today, sir, and even though you gave your time for our webinar. Thank you so much, sir.
Yeah, thank you, ma'am.
I ask all the,
Thank you, ma'am.
thank you, sir. I request all the participants to write your questions in the chat box, and there is instruction for participant. Please fill the feedback form to get the e-certificate. The link is already shared in the chat box, and you'll get certificate within 2 to 3 days. So be patient with that. And if you're facing any problem in filling the feedback form or submitting the feedback form, you can save the link for few hours. We'll keep the link active for a couple of hours, and you can always mail us if you are facing any problem.
Okay, ma'am.
Okay,
Thank
now
you.
moving
Thank you.
ahead.
Okay, ma'am.
Thank you, sir. Now it is time for the open discussion session. I request all the 3 speakers, Aditya Jain sir, Samir sir, and Subrato sir, to switch on your video. And I invite Dr. J.B. Prajapati sir to coordinate this session. And I request Dr. Sujit to share the questions of participants.
Hello.
Yes, sir.
Am I audible now?
Yes, sir. Yes, sir.
Yes.
Yes, sir.
Okay. So, thank you very much, all the 3 speakers. Let us first take the question from the audience who are present.
Okay.
56 persons. If there are any questions, there are
Yes,
questions first, and then questions from the chat box.
I have unmuted the participants. If you have any questions, you can ask. You can unmute yourself and ask the questions to the speakers. If any participants wants to ask, please unmute yourself.
Good morning, sir.
Yes, go ahead.
Good morning, sir.
Yes.
Myself, faculty from Department of Food Technology. So I would like to ask, is there any impact of irradiation or microwave heating or infrared heating on biopeptide quantity or the quality in the food material?
You can, I mean, who are you referring to? The question is from whom you can.
I think
Aditya Jain, sir. Biopeptide.
Sir, can you
Aditya,
get the
please.
question?
I think biopeptides was taken by Dr. Hathi.
yes. Yeah.
yes,
I
sir.
was
Sorry,
thinking why. So your question is that you want to see the effect of microwave heating or other treatments? What other treatments you told?
Infrared heating. Any radiation, sir.
Infrared. Okay. So basically, but what I can understand that microwave heating or infrared. In microwave heating, basically, you are heating the water molecules, okay, for heating the particular products. But it depends only on the how much heating is done, because the products which we prepared in our lab from the mill, we basically do the sterilization. It is around 121 degrees Celsius, okay, for 15 minutes, and then we prepared the product through fermentation. So if you are doing microwave heating or infrared for preheat treatment of that particular milk or whatever the food matrix, I don't know. So you have to do the trials that what kind of changes are there. But in sterilization, what we have done here, there is no any kind of problems. But we did not do work on microwave heating or infrared.
So if time permits, I have one more question, sir.
Yes, please go ahead.
So you have given a slide that there are biopeptides available non-dairy products also, like plant-based and some cereal grains as well as vegetables.
Whey peptides?
Yes.
Not you can add the whey peptides in any food formulations. Some of the whey protein hydrolysates are available, or you can prepare in your lab or other way, and you can add into your any kind of food matrix. Okay. Plant protein, it is a whey protein is basically from the milk. It is a milk protein. It is not from the plant protein, but in plant foods, you can add it. I have given one example where they have added the whey protein hydrolysate as an ingredient into the plant foods.
Thank you, sir.
Actually, all these bioactive peptides can go into fast ingredients and as supplement.
सर आपका आवाज क्लियर नहीं आ रहा है
Sir, your
जेबी
voice.
सर,
Yes, you're losing your voice, sir.
आप क्लोज हो जाइए सर माइक के पास जेबी सर.
Also, can I request Mr. Saxena sir to stop sharing your screen? You can stop sharing your screen, Saxena sir.
Can you stop it from there, because I am not. So suppose second I left actually. I am representing.
We'll try.
नॉइज है सर.
Hello. Is it clear now?
Yes,
Subrata?
yes.
Yes sir, yes. Now it is. Now you said it.
Because computer shows 2-3 options and then I am confused.
no, sir,
Anyway, so bioactive peptide is a big market. They are being sold in different for different applications. I think, but I have one question for Swami Rishi here. Swami, I don't see probiotic food from Amul side. Is there any plan to highlight for generic benefits, but for special health claims also?
Sir, your voice is not very clear. I could not get the questions.
There is a lot of disturbance.
Voice.
Kafi disturbance. I just want to say, as the marketing strategy for Amul's probiotic products.
Sir, the basic strategy for Amul probiotic products was to provide the benefits to the masses. So that's why the most consumed or what we can say is the most popular product, like buttermilk, was chosen as the vehicle to drive the probiotic into the masses, so that everyone can consume the probiotic and take the benefits of the probiotics at a very nominal price. So that was the basis for which we focused on buttermilk and was considered as the vehicle to spread this probiotic.
That is excellent, but that is not the answer to my question. Actually, as Yakult is advertising with some stars that it is good for your gut and Amul is not focusing something on probiotics. No doubt, there are many products with probiotics. Many people do not know even. Say Amul Chocobar is with probiotics, but the consumers do not know that it has got live probiotics in that. So there should be some awareness campaign about the probiotics in your products.
Agree, sir. But there are a few FSSAI legal obligations that certain things cannot be mentioned on the packs. So we are trying through the fermented line, because the fermented line is a slightly easier way to promote this rather than these specialized products like chocolate and ice cream. So that I think that will help, because the entire probiotic category will benefit in that way, which will help to rise the sales of say probiotic chocobars and chocolates and things like that. There are some legal actually obligations.
Only FSA requirement, as I told yesterday, is that 10:8 cells per serving size. And that is not a harder requirement to meet. Usually you are adding more than that in even chocobar covers. Anyway, that is the policy. Is there any other question from the audience? I think as Sujit, you opened the mics, there are so many people talking in between, there is a lot of disturbances.
Yes, sir.
So I think you can just unmute the only person who want to raise the hand.
Correct.
Please raise the hand if you have questions, or otherwise you can read it from the chat box and take it.
Right.
Let
Right, sir.
somebody write in the chat box and then you
Sure.
ask them directly from.
Sure. So are there anybody
Go ahead,
from who have raised your hand? Are there anybody who want to ask questions to our speakers?
Yeah, just hello.
Yes.
See, are you? Is it audible?
Yeah. Please identify
Just
yourself.
one question is there. DN Gandhi.
Okay.
from
Please go
Karnal.
ahead, sir.
So I
Yeah.
want to ask a question from Mr. Aditya Jain. Is it audible?
Yes,
Hello?
absolutely.
Yes,
Go
sir,
ahead.
you are very much.
Because you have mentioned it's very good information with regard to regulatory, with regard to all fermented milk products. But in that you have mentioned heat treatment. Although I know it is these products are having short shelf life, but it is not recommended heat treatment, because according to FAO, the fermented milk products, the viable bacteria should be present till it is consumed. Although just to increase the shelf life, you have recommended, and moreover, this heat treatment can also destroy the body and texture of the product and ultimately acceptability also. Do you agree with this?
Yes, sir.
Hello?
That's the question actually. Yes, you are correct that if you heat treat, the bacteria will be destroyed, and the body texture may also be affected, and accordingly, some additives are allowed to be used in the fermented milks heat treated after fermentation. So it depends upon the consumer requirements or the industry requirements. If they want to heat treat and extend the shelf life, it is allowed. It may not be very beneficial from consumers' point of view, but it is allowed, and that's how UHT lassi and UHT buttermilk are available in the market, which are heat treated after fermentation. But you are right that you will not get benefit of bacteria or as much benefit as you would get from consuming product with live bacteria.
But purpose of this adding culture is to take maximum benefits of probiotic or other starter culture, because they are the
Yes.
useful bacteria and they help in the digestion system and other many more. Anyhow, one more suggestion, just a supplement, and those who are working particularly on fermented milk products, starter culture, other thing, to take maximum benefits of this culture, because a lot of now you all know that on probiotic it is a lot of research is going on and lot of advertisement is out there, as in the Amul also, lot of products they have launched, Yakult has also launched, and Mother Dairy also launched, and others are coming. So my purpose with the students, particularly those who are new research worker, I also try to work that you make the concentrated culture in the skim milk or whey also as a base, and make it concentrated in the put in the sachet form, just like a shampoo is there. Buy one, this 1 rupee, 2 rupee sachet. Similarly, different probiotic culture should be concentrated and packed in this sachet. And this should be sold in the market so that anybody can use it for making product also in the at home domestic condition. Then they can also put in the juice, fruit juice, and take the benefit. They can also put in the water. They can take as a lassi also. Like that, this is my suggestion is there. If these can be taken up, and the maximum benefit of probiotic culture can be harvested. This is what I think. Thank you.
This is a very good suggestion from Dr. Gandhi. I think Amul should think about it and let the culture students.
Because it is for everybody. It's a very nice one. Of course, I know the shelf life is less, but it should be, awareness should be there. They should be stored under low temperature, not like shampoo is hanging in the shops, but it should be in the, whereas other fermented milk products are stored under refrigeration. It's a cold chain system, and it should be there. It's very good. I left this project in between. So I wanted that people should continue and should come in the market also. And in that way, you can make any product, whether it is for gastrointestinal disorder or it is culture is for this one cardiovascular diseases. It is good for diabetes also, and many more. Now even cancer also. A lot of people are saying this culture is very good too. So in that way, particularly, it should be done. That will be a good idea if anybody can do it. Thank you.
Yes, sir. Sir, thank you for a good suggestion. Actually, Dr. Srija has also done under her PhD work. She has developed capsules with probiotic cultures into that, and also the dahi cultures, so that it can be used by housewives to make dahi. Just put empty out one capsule in your glass of milk, and dahi will be ready by tomorrow morning. So this kind of innovations are there, but probably our industry is a little bit skeptic to adapt the innovations which the R&D institutions are developing.
Right.
And that is why appeal to BIREC and Deputy that please do something to have some interlinking with the industry, so that whatever innovations which will come from the universities go to the right persons who can take a little bit of risk, because these products are not necessarily ready to go to market.
Yeah, those who are having laboratories or facilities, they can go for it, particularly.
So go, and it will be really useful to be. And we should promote indigenous culture. We should not be dependent on all.
Right.
There are many things available with SMC college also.
Yeah.
So has NDI also. But hardly any industries come forward to take up those cultures and use in their own products. This is the pity.
It's,
Thank you very much, sir.
Thank you so much.
Please post another question in the chat box.
Yes, sir. So if we're done with the live participants, I'll just read out the questions from the chat box. In any case, if you want to stop me and ask your own questions, just feel free to do that. So I'll just go ahead with the questions. So first question is from I think Karthik Bajpai. It is he's asking, sir, why it is required to state contain milk with allergen information for dairy product, considering that these products are inherently derived from milk. This question is for Mr. Jain. He's asking, sir, that why is it required to state contains milk in allergen information for dairy products, considering that these products are inherently derived from milk?
Yeah, because it's a legal requirement and consumer needs to be informed that milk could be allergic to some of the consumers. So that's just a legal requirement. Yes, logically speaking, if one knows that he or she is allergic to some of the food products, and if it's a single ingredient product, one should be able to know that, yes, this is a milk-based product and a declaration may not be necessary. But because it's a sensitive kind of thing that allergic people, if they just consume by accident some product and it's not written on that, and they may face allergic reactions. So it's always prudent to declare certain, things that one is reminded of, that this could be allergic to someone. So that's probably the reason why even if it is made wholly from milk and it's a single ingredient product, allergy information is still to be declared.
Thank you, sir. I think the question has been answered. Next question is from Nazma Haq. So this question is for us only. It's for BIRAC. That she's asking, BIRAC, does it gives any kind of funding for startups, companies who wants to manufacture probiotic beverages? If yes, can we apply, and do they cover infrastructure, manpower, equipment, etc.? So like I said yesterday also, BIRAC supports startups, entrepreneurs, companies, industries through its various schemes. So please visit BIRAC website for information. We support ideation to early stage validation to late stage till commercialization. We do the handholding through our different schemes like BIG, SIBRI, PACE, and BIPP. Please visit our website, and if you have any queries, please mail it to me if you want. And also, if you want to collaborate with our organizer for the webinar today, SMC College of Dairy Science, I request the industries to please reach out to them. If you want to go have a further collaboration with SMC College of Dairy Science, you can do that. But however, you should be very transparent with the kind of technology you are going to share, and also make use of the expertise that we have in SMC College of Dairy Science. So that's the one more question is, sir, from again Karthik. I think this is for Dr. Hathi sir. He's asking you, sir, that as recognizing the infant stage of the bioactive peptides market in India, is there substantial potential for its success in the Indian context? And if so, how can we as students actively contribute to its growth?
As I have already said, that some of the countries already produce the bioactive peptide-rich fermented milks. Okay, but again, I will tell you that it is a functional foods or healthy food, so clinical trial is very much important. When you will say that because these bioactive peptides you are adding or preparing some kind of health benefits you are asking for, so you have to go for, you can make a product, but proper validation is required in Indian market and any market. So you have to take care of it. You can produce, you can have, if you have a technology or idea, you can go for it. But again, I will suggest that clinical validation is very much important. That how much peptide should be present in a particular dose, as I have already discussed in 2-3 in vivo studies. And I have given the example of Calpis. They have done a clinical trial on antihypertensive. Okay, and they have quantified 2 peptides, IPP and VPP. So your product should have a very clear-cut health beneficial claim with the clinical data. Then you can go for a product preparations.
This is one more question for you in the same area from Dr. Haq from IIT Delhi. He's asking you like there are some stability changes with the use of bioactive peptides in functional foods or drugs. So, like scaling up the production often results in sometimes irreproducible peptides, and unguaranteed quality stability might hinder the commercial production of bioactive peptides. Also, the most convenient and acceptable delivery method of bioactive peptide is oral administration. However, it has been seen in few articles as well as in some research papers that gastrointestinal digestion and rapid metabolism in plasma, liver, and kidney with short half-lives greatly affects the efficacy of bioactive peptides in vivo. So, the application of bioactive peptides sometimes is greatly hindered if high doses and frequent administration are required to ensure efficacy. What are your thoughts on this?
Yes, I got his concern about the bioactive peptide stability. Basically, he's asking about the bioavailability and biostability of the peptides in the GIT tract. When I presented the topic, I said that the peptides which are having a gastric indigestion resistant, that can be quantified, that can be characterized, because all the peptides cannot be used for product formulation. Some of the peptides you have to screen out. You have to do some in vitro digestions in your lab that they are able to resist these stomach conditions, because the pH is less, the lot of proteolytic enzymes are there. When they will reach to the intestine again, other proteolytic enzymes will come and they will degrade or hydrolyze the peptides. So some of the amino acids are there, hydrophobic amino acids there, aromatic amino acid residues are there. They can resist these digestion conditions in vitro, even in vivo. So you have to screen out in vitro digestions. You have to develop a model in your lab. You have to test it that these peptides can tolerate these kind of conditions. Then only you can go for the in vivo trial. So first you have to develop a model, in vitro digestion model, where you can test that these peptides can be tolerated these conditions, and then only you can go for in vivo. There is a big concern about the biostability and bioavailability of the peptides. But again, I am telling you that those peptides already being characterized, it is being available in the BioPep database. Those who have already given some trials on in vivo, you can take their reference, or you can follow their protocols for clinical studies also.
Thank you, sir. I think Dr. Hathi has already answered your questions, and hope you're satisfied with that. Okay, sir, he has written to me that he's thankful for your brief explanation.
Okay.
And there are also a lot of good comments regarding three of our speakers today. Like, people have benefited from the 2 days webinar series also. So they're congratulating all the speakers. A lot of feedbacks has come to us. And also especially to everybody, including a special thanks is for Dr. Prajapati sir, because it seems few of them wanted you as a speaker also. So, and few of them have heard you during the last webinar in Nehu. So they're really happy to have you on board for the 2 days. So they're thankful to the team to have you in the board. One more question.
Before you conclude, I just
Yes,
want to remind of months.
There are a few questions are there, 2, 3. Can I go ahead with that?
Yeah, please go ahead.
With your permission. This question is for 3 of the speakers today, Mr. Saxena, Mr. Jain, and Dr. Hathi. That are there any bioactive derived peptides that are available in India, and what would be the regulatory clearance required for the same, FSSAI or FDA? Which agency would have to be approached for the clearances?
So first, I am telling about the bioactive peptides available product in India. I think there is no any product available in Indian market. There is a big scope for us to develop, and the regulatory part, Prajapati sir and Jain sir will tell about the FSSAI.
No, Aditya, please reply to that.
As of now, there is no specific guideline for permissions, but yes, it will be treated as a novel ingredient, and hence it will have to go for specific approvals. And depending upon the type of ingredient, the guidelines to be followed or specific protocols to be followed and demonstrated, they will be identified by the FSSAI through their panels, and then permission can be given.
There are some imported bioactive peptides are available in pharma stores, but nothing from local industry.
Right. Sir, one more question is for Amul, to Mr. Saxon specifically, that what are the challenges faced during production and storage of probiotic ice cream, and which strain was used for probiotic ice cream production, and what is the response of the customers in Anand as well as in other states when compared to non-probiotic Amul ice creams? It's from Dr. Chandrakala Sharma.
Yeah, probiotic we are adding, as Samir sir had specifically mentioned, that we have started from 2007. So Chocobar probiotic we had already started, but the strains which are added into that are two strains. One is the LA05, Lactobacillus acidophilus, and the second strain is Bifidobacterium animalis, subspecies lactis. These two strains are added into probiotic Chocobar. And to be very frank, comparative studies have not been done. We are our entire Chocobar range is probiotic. So I'm not sure about. I'm really sure that most studies have been done. Comparative studies have been done, but shelf life, regarding the shelf life and the temperature conditions, minus 18 degrees centigrade is the storage conditions which are recommended for this product, which has to be maintained throughout the.
Thank you, sir. I think the question has been answered. One more.
Just to add to that,
Yes, sir. Please.
ये जो Chocobar है, they ensure they do in the beginning that this enough cell numbers of BP12 and Ali5 are there, and probably they have ensured that till the end of shelf life also the retail. As far as the health benefits of these two cultures, if you want to know, you can go to the website of Krishi Nansen, and they have given a lot of literature on these two strains. Even though Amul has not conducted clinical trials to prove health benefit, and that is why they are not claiming these bacteria has got these kind of benefits. Because as per the requirement of DPT ICMR guideline, if you want to show that product is having this benefit, then the trial has to be taken on that product. That means if it is in probiotic Chocobar, then Chocobar should have been tested for this kind of benefit. And that is why that kind of work is not repeated here. So anybody who want to know the health benefits of BB12, that is Bifidobacterium animalis with date, and the Lactobacillus acidophilus Ali5, a lot of research papers on these two strains are available on the website of Krishi Nansen.
Thank you, sir.
Thank you.
One more question. It's for me only. It's for Birac. I'll take this up. So this question is like they want to know the elaborative role of Birac in promoting the industry-academia interaction. So I'll just tell you. Thank you for your question. It's Dr. Swami from IIT Madras. So see, Birac is a industry-academia interface, and it's a not-for-profit section. Section 8 Schedule B, set up by Ministry of Science and Technology. So we help in fostering public-private partnership for innovative product technology development, and there are several schemes that we support from ideation to commercialization, and we implement its mandate through a wide range of impact initiatives, be it providing access to this capital through targeted funding, technology transfer, IP management, handholding schemes that brings the innovation excellence to the biotech firms and make them globally competitive. So that's our brief role. For further information, you can always write to me or anyone in Birac. So I think sir will take up one joint question, the final questions for the day. That is, which source of bioactive peptides are having better absorption in human digestion, dairy or non-dairy? And are there any bioactive milk derived peptides? That is a repetitive same question. Same question has been asked again. And the production of bioactive peptides which are restricted by the availability of substrate. So
okay. So their first question is that bioavailability, right?
Yes, sir. Bioavailability of the bioactive peptides.
Or in dairy matrix or other food matrix.
Yes.
Is the absorption of the peptides, right? So again, the same thing I have already discussed, that the length of the peptides is very important. If it is because when it will get absorbed, they will absorb through different transporters. When it will reach to the intestine, because intestine is the place where these molecules are getting absorbed, even in your nutrition also. So the peptides which are di or tripeptides, there are 2-3 transporter systems are there. So small peptides easily absorbed, but again I'm telling you that the transporter should get signaled, and they will have to take off, and then they will pass through the epithelial lines of the intestines, and then finally it goes to the blood circulation system, and it will goes to the target organs. Okay, so bioavailability totally depending on the sequence of the or the length of the peptides, and again I'm telling you that how much concentrations the peptides are reaching to that particular place. That is very important. Means quantification of the particular peptides which are giving through different doses or through food matrix. Maybe it's from milk or from other food proteins. It doesn't matter that the fermented milk will carry more peptides or other food proteins or not, because you are very much particular about the particular target peptides. You have to select that which peptides you want to get is enhanced or enriched. Okay, that peptides you have to enrich with the product, and then you have to deliver it to different food matrix. So the peptide length, peptide sequence, and quantity is very important for the bioavailability and biostability also. And one second question was what?
The second question is, yeah, so the second question was, is the production of bioactive peptides are restricted by the availability of the substrate?
Not that. If you see the recent literatures, the peoples are extracting the peptides from the alternative protein sources, like the fins from the fish, means waste materials they are looking for from agriculture or animal waste, or the fish market, the waste material which are rich with proteins, and they are extracting the peptides, because you are only purifying the peptides, then you can use anywhere. So it's not restricted to any substrate. If you are using the waste material, it will get more value added product. So there is no restriction on the substrates. But main thing is that what kind of peptides you are targeting and which quantity you are targeting, and their characteristics. That is important.
Thank you, sir. One more question, actually, just now it has come for Mr. Aditya Jain. So the question is, if the number of viable organisms is not applicable in case of a product subjected to heat treatment, how industry ensures that these products are properly fermented, or it is probiotic product? Is an increase in acidity the sole indicator? This is from Ms. Vandana question.
Yeah, actually heat treatment is allowed only after fermentation. So before fermentation, of course, you have to heat treat the substrate, then you have to go for fermentation, and then if you want to increase the shelf life, you can heat treat so that the bacteriological count will go down and you will get an extended shelf life. So there is no criteria that is applicable for viable bacterial count as long as the heat treated fermented meal goes. When it comes to claims, making claims on probiotic properties, there are products in the market where probiotics can be added after heat treatment also. And then those probiotics, because they are not starter cultures, they will not grow in the product, but they will remain there. You have to ensure that they are delivered in the proper way. They can be microencapsulated or so, and then they will go to the remain in the product unreacted form, and they will go to the intestine and do their job. So it is possible to have probiotic benefits even after heat treatment, but then there's a technological requirement that they have to be delivered in a particular fashion. But yes, the other bacteria where there is no claim of probiotic, such as starter cultures, etc., they will not be available to the consumer in the numbers, and there is no claim made on that. So to that extent, the fermented milk will be an intermediate kind of product where you will get the benefits of fermentation because the product indeed has been fermented. There have been all the changes that take place during fermentation have already taken place. It is only the bacteria that is being destroyed. And bacteria, the starter bacteria is not claimed to have any benefit. So that way, there is a fine distinction that it is not so that all the starter bacteria will give you some benefit in the gut. So even after heat treatment, the benefits that you get after fermentation of the product will remain.
Thanks. Thank you, sir. I think all the questions have been answered, and if you have other queries, you can mail that to us if you have any more queries. So a few of the participants were writing to us that the feedback link is not working or not currently accepting the submissions. We request you to apply after some hours again. It is active. Maybe the limit has been reached for today, but however, we assure you that you will get in the due time if you have participated for 2 days. So only mere registration will not give you a certificate. You have to be an active participant for the 2 days the webinar has been conducted. So you have to be patient and be kind to the organizers also. It needs some time to frame some. Maybe the limit has reached for today. So we ask you to be patient on this. You'll get it. So that's all. I think over to you, sir. If anybody has no questions from the participants, I'll hand the session to JB, sir.
Yeah, I think we had enough good discussion. Just to conclude, I have 3 suggestions, rather 3 reminders. Number 1 is that as there is a lot of enthusiasm from so many institutions, we can see more than 1,000 attendees and more than 100 here on this WebEx also. There is one thing which is lacking is systematic training for biomanufacturing of probiotics. And SMC Club Dairy Science under the leadership of Dr. Srijan Subrat and Dr. Mittal have good leadership on this. So I think BIREC should organize such training programs at this college. I am also available here throughout. And also we have a plus point of Vidya Dairy, where we manufacture so many probiotic products also. So we can have a pilot scale study, practicals also. And we can also exposure here to Amul and ADDP and other things. So Anand SMC is handling these things. I strongly recommend that BIREC should have organized training program for this academic for such 3 days, 5 days, 7 days. Second suggestion is that BIREC should organize one industry academia meet, because there are so many research outcomes from the academic institutions, especially with respect to dairy cultures. We have so many things from college, India, NTTP, and other institutions. That relevant industry come in contact, direct contact, and BIREC can become as a mediator for transferring the technologies. And the third suggestion which I recommended last time, that we have a lot of biodiversity in northeastern India. There are many cultures, most of them are non-bacterial, yeast cultures, mold cultures. And there should be one reservoir of such cultures in the northeastern part. And probably NEHU, under the leadership of Dr. Mishra, has done a lot of work in Meghalaya. So BIREC should think of setting such center of excellence for preservation of northeastern India's at NEHU. So these are my 3 recommendations. I hope this will be taken positively by the DPT and by BIREC. And I'm thankful to all the 3 eminent speakers who have presented their perspective, and all leaders in their own field. And we are happy that BIREC is taking such initiatives, because these are total 4 days which was spent on talking about the probiotics and biomanufacturing of that. Probably we have to have one high-level seminar on genomics of probiotics later on at some other place. So thank you very much. Thank you, BIREC and DPT. Thank you, SMC College and Microbiology Department for getting into this program. And I'm also highly benefited by the presentations, even though I could not fully listen to any of the presentations. But I stand for questions. Thank you so much. Thank you. Over to you, Sri.
Thank you.
Sir, just one thing.
Thank you, sir.
Just one thing to add, JB sir, thank you for your recommendations. All the 3 recommendations will be taken ahead, and we're also working on that. We expect a proposal from SMC College Dairy Science to work on the hands-on training or capacity building. Then we can move ahead, since we have Amul and Vidya Dairy also. So we can go ahead with that. Also for Northeastern, we are working on the biomanufacturing part as agenda and initiative of DBT. So we are also on the board. Hopefully something productive will come out soon. And having you is always an honor for BIREC and DBT, always having you on the board, sir. And I'm really thankful to the head, Dr. Srija ma'am, Dr. Mittal, and the coordinator, Dr. Hathi sir, and all the speakers who have given their time for the last 2 days for benefiting us and all the entrepreneurs, students, researchers, companies, industries. So we are really thankful who have patiently listened to us and been a part of this 2 days webinar series. So thank you all. Hopefully we'll come up with some new initiatives in this area very soon. We're working on that. Thank you. Over to you, Dr. Amitabh.
Thank you very much. We are in fact looking forward to collaborate with you. Thank you, JB Prajapati sir, for conducting the session smoothly and concluding the open discussion session. Thank you all the speakers for answering the questions of participants. Thank you, Dr. Sujit, again for sharing all the questions of participants. So here we come to the end of second day of the webinar. I'm here to express the vote of thanks. I would like to thank everyone on behalf of organizing team for the successful completion of 2-day webinar on biomanufacturing of probiotics and fermented foods, a step towards health status and social well-being. I would like to start thanking our great sponsors, Biotechnology Industry Research Assistance Council, BIREC team, a public sector enterprise set up by Department of Biotechnology, DBT. The members are Dr. Shilpi, Dr. Sujit, and others. For trusting us and providing the opportunity to conduct such a webinar second time, we have already conducted one webinar in January 2024. I would like to thank our principal sir for his constant support and for being motivational force for us to conduct such functions. I also would like to express my heartfelt gratitude to the invited speakers for accepting our invitation and sharing your invaluable experience with the participants. I'm sure that everyone is going with some key message from these 2 days deliberation. At last, thank you so much, dear participants. Without you, this program is not possible. Thank you everyone. Have a great day.
Thank you.
Thank you. Thank you.
Thank you. Thank you so much.
Thank you very much. Goodbye. Have a great day.
Thank you, sir. Thank you so much. Thank you, everybody.
Thank you.
You are talking.